Sodium potassium ATPase sodium transport. Sodium together with water is eliminated from the cell by an active transport mechanism generally called the sodium pump. This mechanism, discovered by Hodgkin and Keynes in 1955, was soon associated by Escow with the enzyme sodium potassium ATPase. This enzyme is able to couple the hydrolysis of ATP with the removal of sodium from the cytoplasm against an unfavorable electrochemical gradient. Sodium potassium ATPase is an integral membrane protein that needs to be released and isolated. The best purification have been obtained from tissue rich in enzymes such as electric yield organ, shark rectal gland, salt gland of marine birds, and kidney of mammals. These purified preparations treated with negative staining show fragments that contain a granular structure on both sides of the membrane. Fridge fracture particle of 10 nm have a smaller subunit in a quadripartite structure. The molecular weight of oligomer would be greater than 50,000 Dalton. The sodium potassium ATPase has been isolated in pure form from the kidney and reconstituted in phospholipid vesicle. Localization of sodium potassium ATPase in tissues can be done by subcellular fractionation autoradiography with 3H oubin and the use of labeled antibodies. With these cytochemical methods, it may be shown that the enzyme is localized almost exclusively at the basolateral side of sodium transporting epithelia, such as the thick limb of kidney distal tubules. This localization suggests that sodium which enters through the apex of the cell driven by the concentration gradient is actively transported to the extracellular space at the basolateral surface of the cell. In case of red blood cells, it has been estimated that there are about 5,000 ATPases, each of which are extrude 20 sodium ions per second. Sodium potassium ATPase within the red cell membrane. Hydrolysis of 1 ATP provides the energy for the linked transport of 2 potassium ion towards the inside and 3 sodium ion towards the outside of the cell. This diagram shows the vectorial characteristic of the enzyme, which are sensitive to ATP on the inside of the membrane but not the outside. This ATPase is stimulated by a mixture of both sodium and potassium and is inhibited by a cardiotonic glycoside oubin. These specific inhibitors bind with the enzyme only from the extracellular surface and can be used as a marker and to localize and measure the number of enzyme molecules in a membrane. Sodium potassium ATPase can be inhibited by Vendette, but this inhibitor acts on the cytoplasmic side of the enzyme. The transport reaction takes place in two major steps. Step 1 consists of formation of a covalent phosphoenzyme intermediate on the inner side of the membrane and in the presence of sodium. This first reaction is inhibited by calcium but not by oubin. In step 2, the sodium enzyme phosphorus complex is hydrolyzed to form free enzyme and phosphorus inorganic. This reaction requires potassium added to the outside of the membrane and is inhibited by oubin, which completes with potassium. Step 1 and 2 can be represented as follows. In presence of sodium plus ATP plus enzyme, sodium plus enzyme plus phosphorus or release of phosphorus or ADP. Sodium enzyme phosphorus plus potassium, sodium out, potassium in, phosphorus inorganic is used or enzyme is released. It may be observed that sodium and potassium are ultimately translocated in the opposite directions from which they were bound. In the sodium-potassium ATPase system, the transport of sodium outward is compensated by that of potassium inward and electrical neutrality is achieved. However, there are other cases in which the pumps becomes electrogenic since the eject of sodium is not compensated by entrance of potassium. The electrogenic pump generates a potential that may provide the driving force to transport other solutes. Several substances, glucose, amino acid may enter the cell by means of the sodium pump.